wanted to revisit CRISPR and Cas9 just for a brief second. The way that this works, and I didn't state this very well yesterday, the Cas9 is something that will target a sequence of DNA based on the mRNA or the RNA that's inside of it. So researchers are able to take an RNA sequence that relates to a gene that they want the Cas9 to bind onto, and then they stick the RNA sequence into the Cas9 and it will attach to that portion of the DNA. So we can direct Cas9 with the CRISPR-Cas9 method to what portion of DNA it should cut based on the RNA that we put into it. Okay, so that can be used to either knock a gene out and turn it off, or we can use it to try to get the cell to repair a specific gene in a certain way if we give it a complete copy or a correct copy of the DNA to repair from. Okay, so that was that section. GMOs, okay, genetically modified organisms.
About this yesterday.
Huh? We talked about it yesterday,
I
or
wrote a paper about it.
you wrote a paper about it?
Yeah, we had to write an in-class essay.
Oh, what class?
English.
Oh, nice. So did you know, were you given articles to read about it beforehand, or were you just like writing during?
That's how it goes.
Okay, okay, all right. So you guys might know more than I do about some of the ethical or the debates that people have about it. So if you have a comment that you want to add, feel free to, okay? But briefly, genetically modified organisms can come in different types. So we can look at just cells that are genetically modified, or we can look at animals and plants as a whole that are genetically modified. So a lot of the time we use the cells to produce products like proteins that we can't get from a natural source in large amounts. So that means that we take plasmids through the method from 12.1. We insert a gene that we want to be produced to make certain proteins in the plasmid. We put those plasmids in bacteria. The bacteria make the protein. Bacteria cannot make proteins for everything that humans make, though, or everything that eukaryotes make, because they're prokaryotes. So there are some things that they can't do. For example, they don't edit proteins very well. So a lot of proteins for humans or eukaryotes require taking the amino acids, that protein sequence, and adding a carbohydrate to it. So adding a sugar to it to make a glycoprotein. Bacteria don't add sugars the right way for eukaryotes. So if we want to mass produce human proteins, if it's a glycoprotein, we usually have to use a eukaryotic organism to do that. So then that brings in yeast. We talked about using yeast for brewing, using yeast for baking bread and things like that. We also can use yeast, because it's a eukaryotic organism, to produce some human proteins. So an example of that is found later in the chapter, so we'll see a picture of it. But we used yeast to make insulin, okay? And I haven't had this conversation yet with my child's daycare worker, but she takes insulin. And I'm interested to see if she knows that she probably gets it from a genetically modified source. I don't know if she really cares, but it's an interesting question just to ask because of some of the other conversations I've had with her about stuff. So, what's up?
Your child has diabetes?
No, the daycare worker has diabetes. No. No, thankfully Anna doesn't have diabetes. Hopefully she'll never develop it. Yeah.
That's not sad, that would be sad.
It would be sad. And it would be sad. Bacteria are used to help make different kinds of drugs, insulin, growth hormone. There's things to treat specific kinds of cancer that we use bacterial cells for that have been modified. Okay, so it's used for a lot of different purposes. If you have had all of your vaccines and you're up to date on those, you probably had a hepatitis B vaccine that was made using recombinant DNA. So it was produced using this method. Okay, so that's just interesting. Okay. Sometimes we have to use mammals in order to have them make the protein that we're looking for. So this goat is a wonderful example of this. They inserted modified cells into the goat. So the goat was already alive and they inserted these cells, which is different than cloning the organism or creating an embryo that has the modified cell in it and letting it develop. Okay. But this particular goat makes a human blood protein and it produces it into the milk. And then from that milk, as we milk the goat, we can collect the protein and then give that protein as a shot to someone who needs it. So it's maybe kind of a roundabout way of getting to it, but if something has to make the protein, this is one option for that. Okay, the most expensive option is what's described here, and this is where you take a whole animal. So you take an embryonic cell or a zygote that you've modified, and you stick that into a surrogate mom to have that embryo develop into a baby. And if that embryo develops correctly, then you have an animal that has that genetic change inside of its DNA. So then at that point, once that's developed, we can try to clone that animal. And then you get a whole like herd or farm of genetically modified animals that have the gene that you want modified in them already. Okay. Again, that's the most expensive option because cloning is expensive. It's not always successful. Clones die earlier.
Aren't they just like breeding?
So why don't they allow sexual reproduction to take place?
Yeah.
If you do that, then you might not get the correct version of the gene that you want expressed in the gametes. So yeah, genetic variation. Yeah. Okay. Now you could do that, and I don't know if that would end up being cheaper than trying to clone them. Yeah, it's the Punnett Square stuff, which we're going to go into next. That's our next unit. Okay. After this. So yeah.
Is it mitosis or meiosis?
Meiosis would be the one that produces gametes. Mitosis is producing identical cells.
Yeah, I don't like
Meiosis, meiosis, either way works. Either way works. Okay. So DNA tech. This is some more specific examples of how we use biotechnology for treating diseases and for making pharmaceutical products. So insulin is mentioned again. Human growth hormone. This is something that some people with certain types of dwarfism lack, so they don't make in their bodies correctly. We can treat that by giving them shots of human growth hormone. Okay, so some types of dwarfism or some types of certain diseases are fixable, I guess, if you want to put it that way, because we can provide the protein or the hormone that's missing. And so humulin is the name of the insulin that's made by bacteria. The growth hormone is something that we can give to some people, and that's a lab-made gene. So in this section so far, if you've been doing the reading, you've seen E. coli a lot. E. coli is also in the news because it causes like lots of diseases. So, Splash House? I didn't know that. I missed that news story. But E. coli is the name of a group of bacteria. There are different strains of that bacteria. So the E. coli that are used in the lab to produce medicines and things like that are not the kind that cause disease. So as far as from that aspect, they're safe. You're not going to get food poisoning or sickness from those kinds of E. coli. But the kinds of E. coli where they recall all of the spinach and romaine at the store, that's the kind of E. coli that would cause food poisoning. Yeah, it is sad. Okay, I remember that happened around Thanksgiving this year, and people were posting around the meme that what was it? The pumpkin pie is safer to eat than the salad. Like, you know, yeah, it's true because they recalled all of the salad. Diagnosing different diseases with DNA technology, we're able to take a DNA sequence from someone and figure out if they have cystic fibrosis. We're also able to figure out if they're a carrier for cystic fibrosis or hemophilia or dwarfism or any of hundreds of other different diseases. So if you know that you're a carrier for something, that allows you to make different life decisions. Some people want that power and some people don't. Okay.
So if you have like dwarf parents, but you're not a dwarf, well, can you have dwarf parents and not be a dwarf?
That would depend on what type of mutation the parents have that
Like
causes the
different kinds
dwarfism. There would be different kinds of dwarfism. About that? Okay, yeah.
It's like you can be a normal person and have a dwarf. But yeah,
You
that's
could be,
obviously
yes,
true.
yes, yes.
But if you're a normal person, do you carry the dwarfism stuff?
You can, because I believe most of the mutations for dwarfism are recessive alleles, which means you can be a carrier for it and not show it. You could pass it on to your kids.
What is it?
Hmm?
What is it like?
A dwarfism?
Yeah.
The technical
I know, like,
the technical term for it's achondroplasia, and
like
it's
how do you get it? Like
There's a couple of different mutations. So the
Absolutely,
mutation
they
that's
all
talked
look the
about
same.
here that's treated with human growth hormone is a mutation in producing the hormone. So it just means that your body doesn't produce the hormone that causes you to grow. So you don't grow as much as you normally
Why
would.
do they all look the same?
Why do who all what?
A lot of dwarves look the same. They all have the same like head shape and like
Yeah,
facial
why is their
structure.
head
Okay, so
shaped?
pause for a second. I'm going to go back to cystic fibrosis. Okay, because the video talked about cystic fibrosis. Francis Collins found one of the common mutations. So Francis Collins in the video, when they were talking about cystic fibrosis, they said he discovered the main mutation, but then they also said there's like 1,800 other mutations in that gene that can still cause cystic fibrosis. So there's more than one change in a sequence of DNA that can make a protein not work. Okay, so that's part of the difficulty when we talk about humans, because generally most of our traits are polygenic. So the things that our bodies are able to do are controlled by more than one gene. So you can change this gene or gene 2 or gene 5 and still have the same process not work anymore. So it's going to cause the same symptoms even though the underlying reason is different. It's a different mutation.
So
Okay.
2 normal people could have a dwarf.
Yeah.
Yeah,
Yes.
That's what I was
I
thinking.
didn't
Yes.
know that.
I was thinking about my
That's
cousin,
just like-
his cousin, like both their parents are normal, and then they have a regular child and then a dwarf.
And just like two normal parents can have one child with cystic fibrosis and another child without cystic fibrosis.
Like in the face. It's just
Because it all depends on what alleles are in the gametes. So it's genetic variation. Okay. So we can diagnose different diseases, and at this point we can try to diagnose a lot of diseases in the womb before the embryo finishes developing. And there's pros and cons to that. And then we can also use the DNA technology to make vaccines. So the vaccine I mentioned already was the vaccine for hepatitis B. All the hepatitises, there's A, B, and C, cause different kinds of liver disease. But with hepatitis B, the vaccine that we now make takes a version of the virus that doesn't cause the disease anymore. It just has the antigens or the proteins on the outside of the virus that the cell can recognize, hey, this is the virus, and it sticks those proteins into or onto a cell that we administer with the vaccine. So your body gets a chance to recognize the virus as something that it doesn't want, so it can attack that without you actually getting the disease. That's how a vaccine is supposed to work. You get really a mild form, or you don't get it at all, but your body is able to develop the defenses to attack it if it sees it again. Okay, so that's a little bit about vaccines. More about GMOs. Okay, we have had GMOs in agriculture for a very long time in the U.S. So if you ate something that has corn or soy in it, you probably ate something that was genetically modified. You just don't know it because companies are not required to label products like contains GMOs if it has genetically modified corn or soy in it. So the GMOs, or genetically modified organisms, are sometimes called transgenic organisms because they have a gene from one organism plus the original genome. So there's more than one species or more than one type of animal or bacteria or plant in the same thing. So some people who are for genetically modified organisms will say there's really no difference between the plants that we can selectively breed together. So one example is taking a type of wheat and a type of rye and causing them to breed together. That's not using any of this DNA technology. It's just selective breeding. And that's transgenic because now you have the genome or the DNA from wheat and from rye in a new plant. There's no difference between that and taking a gene from one plant and putting it in another plant. Okay, and we've been doing stuff like that in agriculture for a very long time. As a matter of fact, all of the wheat that you eat now was genetically modified back around when the Babylonians were around, because wheat as we know it did not exist the way that it does today. So it was crossbred from a few different species of plants to get the wheat that we have. That's technically genetically modifying it, just not with the same technology we have now. Okay, so how are plants bred? It's called cross-pollination. So you take the pollen from one plant and put it on the other plant. And sometimes that's successful, sometimes it's not. A lot of it depends on how many chromosomes the plant has. But plants also do something that's called polyploidy. So they might have like 8 copies of the same chromosome. We are diploid, we only have 2 copies of our chromosomes. So plants are just a whole nother animal, but they're not an animal. Yeah, I know, thanks. Okay, so one example of a genetically modified organism that seems to be very helpful is golden rice. So this is a picture of that. As far as I know, you can't actually buy this in the store here. We don't really have a reason to need it here though. Golden rice is modified to have more vitamin A in it than normal white rice or brown rice. So because of that, it means that people who might not get enough vitamin A in their diet are able to eat this rice, and then they don't have a vitamin A deficiency, okay? And how do you test that? Great question. The next section talks about some of the ethics behind testing with humans, because that's always an issue. It doesn't matter how far into the future we go. I think testing on humans is always going to have some ethical debate for it. But again, we've been genetically modifying plants for a long time. Genetically modifying animals, on the other hand, that doesn't happen very often. It's not approved, at least here in the US, for people to eat genetically modified animals. So one example, and I have to look and see if you can actually buy this yet, but in 2015, the FDA approved sale of a salmon that's genetically modified because it can grow to the same size as a naturally occurring salmon, like in half the time. So because it can grow to a bigger size more quickly, people who have fish farms are able to use that fish, and they can produce more fish more quickly. So it's a kind of supply and demand thing. Hypothetically, that's on store shelves. I just haven't fact-checked that for sure or not. But 2015 was a few years ago, so probably if you buy salmon, you may have bought that kind of salmon. I don't know. I have to fact-check that. Okay. Next section, talking about, again, GMOs. So environmental safety, labeling, should we label it, what's that look like, and will humans be affected by it? So this outlines a couple of different studies that were done. Part of the problem is we can do studies with animals that are similar to humans. So pigs are a good example of that. A pig's digestive system looks fairly similar to ours. And they describe a study that was done with pigs. So they fed the pigs a diet of GMO corn and then non-GMO corn. Didn't seem to affect the pigs at all. And they looked at within like a month and like a year, and then the whole lifespan of the pig. Pigs seemed to be fine. But how do you translate that from working on pigs to working on humans? Okay. The example that's given here was done with Chinese and American scientists working together. So they fed Chinese school children, so between ages 6 and 8, which would be our like 1st through 3rd graders, different kinds of diets. So one diet was included a pill that had pure beta carotene in it. Beta carotenes needed to make vitamin A. So they had the pill needed to make vitamin A. They had golden rice in their diet, or they had spinach, which has naturally occurring beta carotene. Okay, so in order to measure the effect that had on the kids, they drew blood and measured the amount of vitamin A that was in the blood. So the spinach made way less vitamin A than the golden rice or the beta carotene. The golden rice made a comparable amount of carotene or vitamin A compared to the pill though. So you can take a supplement that's pure beta carotene to have a lot of vitamin A, or you could eat golden rice. If all you're eating is spinach or other sources like that that have naturally occurring vitamin A, you're not going to get as much as if you eat golden rice. So they conducted the study, they had permission from everybody to do it. The parents of the kids knew, the Chinese and American scientists worked together and planned it together. But then when data was published, people were very frustrated. You used Chinese kids to do this study, and that's not right, and it's unethical. Okay, how do you translate it from animal study to human study? Okay, and realistically, it might have to do with why didn't you study on American kids instead of Chinese kids? That's probably one of the reasons why some officials were upset about the study too. So not only is it a how can you ethically be safe so that you're not hurting the humans that you study on, but it's a political thing too. Who do you study on? So trying to do studies like this can sometimes lead to manipulating groups of people who don't have as many rights or don't have somebody advocating for them. That's an ethical problem too. Okay. Environmental safety, the concern people have with GMOs, if you plant a GMO, what happens if it breeds with some plant out in the wild? Now that gene that you've modified is just out in the wild, and we don't know what that's going to do to other plants or other organisms. What happens? Okay, that's an environmental concern. And then labeling, some people think you have a right to know if the food you eat has GMOs in it. Okay. People who are an advocate for GMOs say, but we weren't required to tell people when we took a breed of wheat and a brand of rye and bred those together. Like, that was selective breeding. It's artificial selection or artificial breeding. We weren't required to tell people we did that. So why would we have to tell people about GMOs? Okay, so the arguments back and forth. Gene therapy. This is something that's been done successfully a few times in humans. So there are some select studies where this has happened successfully. But again, testing on humans has its own set of red tape and ethical problems. So gene therapy looks like taking the gene that you want that's healthy from a normal person, so a healthy individual, and then you get the RNA from that gene. So you go from DNA to RNA. You put that RNA sequence into a virus that's a harmless virus, so you take the teeth out of the virus, but put the RNA you want into it. Infect the sick person's cells with the virus, and then the sick person's cells will have the healthy version of the gene inserted into their DNA, so into those cells. Then you take the cells that came from the sick patient that now have the healthy copy of the gene and put them back into the patient. Okay. In order for gene therapy to work at this point, we have to treat types of genes that reproduce a lot over a person's lifetime, so that the benefit of inserting a few genes that now have the correct copy will multiply as the person gets older. So bone marrow is a good example of that, because your bone marrow produces all of your blood cells, and it has to divide and multiply many, many times over a person's life. So some other examples of this might be skin cells, could be the lining in your intestines or on your stomach. But to do that gets more complicated depending on what tissue you're trying to insert the cells back into. Okay, but we've been doing bone marrow transplants for a while, so this is kind of like doing a bone marrow transplant. You pull the bone marrow out, put the corrected cell into it, put the bone marrow back in. Okay. So there are some examples here of ways that this have been used correctly. So in a time span of 11 years, they were able to cure 22 kids with an immunodeficiency disease. So it's an inherited disease that meant that they wouldn't develop an immune system. If you don't develop an immune system, you're living like in a bubble. So there's a book by, I think it's John Green, where the girl lives in a bubble because she's supposedly allergic to everything. There you go, everything. I'm like, I know the name. Anyway, there are kids who actually have that disease. So if you're exposed to the common cold, you die. Like, it's not just you have a cold, you have a sniffle and a cough for a few days. You probably die. So that's a huge thing. So with gene therapy, they were able to fix that for 22 people. Okay. Other diseases, we've been able to treat some things related to problems with hemoglobin. We've been able to treat things with rhodopsin. Rhodopsin is a protein that allows you to detect light. And in people who have this disease, eventually they get more blind and more blind until they become completely blind, because they lose that protein and it won't function anymore. So they've been able to correct that for some kids, for some people. Okay. But gene therapy, again, has ethical questions that are a problem. At this point, all the gene therapy that we've done will only affect one individual. It's not going to affect any of the kids that they have. So if we're doing gene therapy, do we start to affect potential future generations? Do we modify gametes? Or do we only modify the person who's an adult and only modify tissues that so it won't be passed on to future generations? What does that look like? So one of the videos that we had watched, the Designer DNA, talked about modifying genes being really close to what the Nazis did. I don't know if you remember that line or remember hearing that. but Nazi scientists practiced eugenics, which was trying to control what future generations looked like. And they did that by killing off people of certain ethnicities or backgrounds. They did that by forcefully sterilizing people. They did that by telling people, if you have a kid that looks like this and you have like a certain number of kids, we'll like give you money. So they tried to encourage people of certain types to have kids together. Okay, but eugenics on this level would look like, we're going to try to eliminate Down syndrome by taking everybody who has a copy of the Down syndrome gene and fixing that gene so that you can't pass Down syndrome on. Sorry, that's actually a really bad example because of how Down syndrome is inherited.
No, but they do that.
But they do that by aborting embryos that have Down syndrome.
I like that when I was watching a video about
and-
it, there was like a group of like Down syndrome people like,
Who were talking about it, yeah, and that's
so sad.
the headline that came from Iceland, because Iceland's the one that the country that's leading the charge on that. But the headline said Iceland is on its way to eliminating Down syndrome, and that's because most women who are pregnant get a genetic test that shows the embryo has Down syndrome or doesn't, and if the embryo has Down syndrome, they get an abortion. So
Do they have to?
no, but a lot of people are choosing to do that. Okay, and that's no different than people under the one-child policy in China choosing to abort an embryo that is female. Okay, but that's something that happened and happens in a couple of other countries as well. So is that ethical? Should that be allowed? Great question. Okay, great question. Okay, DNA profiling. How many of you watch forensic crime shows? Okay, a couple of you. Has everybody seen at least one episode of NCIS?
Yeah.
Okay, maybe Abby Shudo. Okay, like love her character. Okay, but DNA profiling is something that having all of this biotech allows us to do. So this is kind of like getting a 23andMe test done. They don't look at the entire genome sequence. They look at like maybe a few different markers. Okay, so DNA profiling works like you take DNA from a crime scene. Could be blood, could be hair, could be semen. So you take that DNA, you amplify it and look at certain sequences in the DNA, and then you can do the same thing from the DNA of different suspects and either prove innocence or confirm guilt. So DNA profiling can be used both ways, and at this point our technology is advanced enough that it's admissible as concrete evidence. So as long as the DNA is collected the way it's supposed to be and the tests are done so that nothing's contaminated, it's used as concrete evidence for innocence or for someone's guilt. Okay? So the next couple sections help explain that. So the first thing you have to know is PCR. PCR stands for polymerase chain reaction. So hopefully you remember the term polymerase. Polymerase, there's DNA polymerase and RNA polymerase. In this example, we're talking about DNA polymerase because we're going to copy sequences or sections of DNA. So the way this works, you take your sample of DNA and you have that cut into different sequences. Okay. Then in PCR, they take the DNA and heat it up a little bit. That causes the DNA to break into two strands, so the hydrogen bonds disintegrate if you heat it just enough. And then they take the DNA polymerase, and as you cool the DNA back down, the polymerase will attach to the two separate strands of DNA, okay, in these primer sequences, and DNA polymerase will build your DNA back up. So we go from having one copy of the target sequence to 2 copies. That's after one cycle. If you do that again, then we go from 2 copies to 4 copies. If we do that again, we go from 4 to 8. So PCR is called a polymerase chain reaction because we're using polymerase over and over and over again to get more copies of the same DNA sequence. So that's called amplification, just like amplification in the signal transduction pathways. Okay. Obviously, I didn't quite finish talking about this, so I will finish talking on Friday or Tuesday.